Welcome to the Neuroblastoma Parent Global Symposium 2020. My name is Lucy Jones and I'm from Neuroblastoma Australia. I'm here today to present the very first session, the speaker. And the very first session of the symposium is called Neuroblastoma Research, a Global Perspective. Now, this session will be held and presented by Dr. Toby Traher. Now, Dr. Traher is a specialist in paediatric hematology and oncology at Sydney Children's Hospital, and he is also a clinical research fellow at the Children's Cancer Institute and does a huge amount of amazing research in the areas of neuroblastoma and leukemia. Now, before I hand over, I'd like to draw your attention to the Q&A button that you should be able to see at the bottom of your screen. Now, you can use that Q&A button to submit your questions. Actually, you can submit the questions whilst Dr. Traher is actually speaking, and I will be actually noting those questions and will be presenting as many questions as I can at the end of Dr. Traher's presentation. So if there is also a question that you particularly like, you can actually click on it and give it a thumbs up. And if a question has lots of thumbs up, it kind of moves up the list, so it helps us prioritize the questions which are the most popular questions. If you can't find your Q&A button, just hover at the bottom of your screen and it should pop up. So I'm going to actually conclude here, and I'm going to hand over to Dr. Traher, who's going to talk to you now about neuroblastoma research. Thanks very much.
Hi everyone, my name's Toby Traher. I'm a children's cancer specialist at Sydney Children's Hospital, and I'm hoping that you can see my slides. Hang on, that might not have worked. Just give me one moment. Let me do that again. Here we go. My apologies about that. So I'm a children's cancer specialist at Sydney Children's Hospital and also a researcher at the Children's Cancer Institute. And look, it's a real honour to be speaking today as part of this Neuroblastoma Global Parent Symposium. And also an honour to lead off. And really, I wanted to sort of focus really on neuroblastoma research as a global perspective. Down the bottom of my slide, you might notice that there's a magpie, and for non-Australians in the audience, magpies are great collectors. And so through this talk, what I've tried to do is actually collect information from all around the world and sort of put it into the talk, really to give you an idea about how important global collaboration and global research has been in terms of neuroblastoma. So in terms of who I am, I'm a children's cancer specialist. I've been working at Sydney Kids since 2006. I'm also a research fellow at the Children's Cancer Institute, and then I've been lucky enough to be involved in a number of neuroblastoma-associated activities, both locally in Australia through our local Children's Hematology Oncology group. I'm also currently on the steering committee of the Advances in Neuroblastoma Association. I'm part of the CERPIN Trials Group for Australia and New Zealand, which is one of the European Trials Group. I'm a member of the Children's Oncology Group, and again, I'm also a member of our local ethics committee. So I wear a number of different hats in terms of treating kids with neuroblastoma, being involved in research for neuroblastoma, but also being involved with trials groups and the ethics process behind research. And so I think from that point of view, I think it's important to sort of think about how important international collaboration and research has been in actually improving outcomes for kids with neuroblastoma, and how it really provides a very strong template for future progress as well. As you're probably all aware, it's a very complex disease and it's got some quite unique features, and particularly in younger children with more favourable biology. Again, neuroblastoma behaves very differently to in older children, and it's got a much more higher chance of regression or going away with minimal therapy. Whereas in contrast, as many of you know, children with higher risk disease have unfavourable biology, and they often have problems with progressive and aggressive disease. And again, there's still less than ideal outcomes, even with intensification of therapy. And so the images I've really got up on the slide really just show some images from a child from a publication almost 20 years ago now, where they've got a large intra-abdominal neuroblastoma, but without therapy, it's shrunk away. So this is the paradox. Some kids need almost no therapy, whereas other children need very aggressive therapy. My entree into international neuroblastoma research was I was invited as a young investigator, as a junior consultant, to go to one of the INRG meetings. And what really struck me there was how important the international collaboration is and continues to be. And so what is the international neuroblastoma research community? Well, it's huge, and it's many, many components. And obviously it takes in patients' families, and it takes in parent and philanthropic associations. There are organisations like the Advances in Neuroblastoma Research Association. There are intergroup collaborations between large organisations, which have come up with standardisation for treatment staging and risk assessment. Then there are translational research consortia, there are trials consortia, there are data repositories. Obviously, there are national oncology societies, individual hospitals, research institutes, clinicians and researchers, government and funding bodies, and pharmaceutical companies. But what's really important is actually how this all works together as a collaborative network. And I think that's really taken great strides to improve outcomes. And I think that's a good place to start in terms of how we think about how to improve outcomes for neuroblastoma. So I just wanted to spend a little bit of time talking about the Advances in Neuroblastoma Research Association, which is an organisation I'm very proud to be part of. And again, this is a publication that Frank Berthold from Germany had put together, which was really just reflecting upon the progress and the advances in Neuroblastoma Research meeting, and how it's been a tool to drive collaboration and networking amongst basic researchers, translational researchers, and clinicians. And the thing which I thought was really good about the way Frank had analysed the ANR data was over 18 meetings since 1975. He'd really then started to look at what are the networks that had formed internationally in neuroblastoma research. So the two figures here are the figures that he put together, and you can see that this is obviously a map of North America, this is a map of Europe. There are lots of dots, but they're also connected dots. And so this is trying to quantify the amount of collaborative research that's going on, but also the links between the centres as well. And so the most important feature here is there are lots of research organisations and institutions involved, but they're also highly interconnected as well. So over those first 18 meetings, there's been almost 4,000 presentations, and they're roughly equally split between basic research in neuroblastoma, translational research in neuroblastoma, but also clinical trials. And so again, it's a meeting that's just solely focused on neuroblastoma. One of the other entrees that I also had in terms of neuroblastoma was also the International Neuroblastoma Risk Group Task Force, and I went to a meeting in 2005 in Whistler in Canada. And this was, again, where I sort of really started. And what really struck me was a coming together of everybody involved in neuroblastoma, trying to make sure that we made changes the way that research was done and thought about to improve outcomes. So one of the really strong features of this is the concept of international harmonisation, so that research that's done in the clinical sphere can be translated to and understood and is comparable to research in other parts of the world. It is also a way of bringing together not only just clinicians and translational researchers and basic scientists, but get them talking together, and so that you can then use all of the information from individuals to help drive importance in basic biology, translational biology, and clinical research. So the original INRG dataset was put together taking about 8,000 patient datasets from a number of trials groups across Europe, North America, and Japan. But this is not a static organisation, it's living and ongoing today. And so they still take patient datasets, so there are now more than 20,000. And so there's also what's called a data commons, which has been put together by the University of Chicago. And it's really a way of actually making that type of data available so that current researchers continue to use it as well. Now, this is really simple, and this is just the INRG staging system. And really what the INRG was trying to do was to look at what is the best way to harmonise the way that we think about clinically. You'd stage a child who's newly diagnosed with neuroblastoma, and to look at what were previous staging systems and improve upon them, but also to make sure that everybody could be using exactly the same system. So although it seems like a very simple idea, it's also exceptionally powerful because it means that if everybody's actually using the same system, it means it's much more easy to compare data, and it means that you can then also drive the progress and the speed of research as well. Again, as part of that, there have been other international consortia that have also been looking at the response criteria. And again, it's the same very basic, simple idea that as long as everybody's harmonised what they're doing, it means you can compare results. And so that I think that's also, whilst it's very simple, it's also very powerful. And so it means that if you look at a clinical trial that has, say, been conducted in North America or Europe, you can actually compare them much more directly nowadays than, say, 30 years ago. So again, I think this concept of harmonisation, it's a foundation piece of work that's really, really important. Standardisation is important. And so if you think about it, the INRG, the International Neuroblastoma Risk Groups, they were about standardisation and harmonisation of the initial staging and workup, the stratification of neuroblastoma, but also many other aspects about the clinical workup of children with neuroblastoma as well. The International Neuroblastoma Response Criteria are also built around the concept of standardised response assessment. And again, it really is this underpinning of international comparability of results. And so again, the dominant themes that have sort of really happened over the last 20 years in neuroblastoma in terms of clinical research have really been around in non-high-risk neuroblastoma, the ongoing realisation that, again, the biology of this disease is generally favourable and that children with non-high-risk neuroblastoma generally don't need much therapy or can have reduced therapy compared to historical standards. In contrast, for high-risk neuroblastoma, again, there's been a realisation it's an aggressive disease and that despite intensification of therapy, we still don't achieve outcomes that we would like. And so again, the dominant theme in much clinical research in high-risk neuroblastoma is about escalation of therapy, finding new pathways, new biology that we can leverage for treatment, and then the introduction of new agents into therapy. And so there have been successful introduction of anti-G2 antibodies. There's also been the introduction of ALK inhibitors and also the use of, say, high-dose MIBG, which is some of the current trial questions going on at the moment. So if you look at also what's happened, say, in the last 20 years, again, I think it's important to also recognise that there have been a large number of quite successful trials that have actually been undertaken in many different disease settings, non-high-risk disease, high-risk disease, relapsed refractory disease. And I think they're important because not only do they give us information about how to take the field forward, but they also influence standards of care for treatment all around the world. And so in the next couple of slides, really what I've got is just some data and information around clinical trials that have been quite successful. It's not supposed to be all clinical trials. It's basically some of the important trials that have been conducted over the last 20 years. And so if you look at non-high-risk neuroblastoma, it's been very clear from trials conducted in both Europe and North America. You can see spontaneous regression of localized infant neuroblastoma. It's also been very clear that infants with metastatic neuroblastoma often have excellent survival. It's also clear that for children with what's called intermediate risk neuroblastoma, you can have excellent outcomes with ongoing de-escalation of therapy. And so there have been a number of very successful trials consecutively performed by the Children's Oncology Group in that. I think the other thing that's also really important in terms of clinical research is that there are also really important patient populations in neuroblastoma as well. And so there's a very rare syndrome called the dancing eyes, dancing feet syndrome, opsoclonus myoclonus. And again, the Children's Oncology Group have been able to successfully do a randomized trial looking at the introduction and the use of gamma globulin for patients with opsoclonus myoclonus and neuroblastoma. So this is a very rare sort of syndrome associated with neuroblastoma, but it also highlights the possibility that by international sort of thinking and collaboration, you can actually do trials in this these groups of patients. The other thing that I think is also really important is there are a small number of children with neuroblastoma who have intraspinal extension of their neuroblastoma. And there's some important retrospective registry data that, again, the German group published, which showed that although these children who have intraspinal extension, if they're symptomatic at diagnosis, although they might have good neuroblastoma outcomes, they actually have quite poor functional outcomes from the perspective of their neurology. And so from that perspective, again, I think it's a really important sort of study to show that there are still a small subgroup of patients with non-high-risk disease who might have poor outcomes because of specific features of their disease, either opsoclonus myoclonus or spinal canal involvement. In high-risk disease, again, there have been a number of very successful clinical trials that have been done over the last 20 to 30 years. And again, they're very influential, not only to design new trials, but also to influence standards of care as well. And so again, there's been a strong focus on consolidation or high-dose consolidation therapy, and that's been shown to be successful. In Europe, they've done randomized trials to compare what is the best conditioning regimen for a single transplant. In the US, they've done a successful trial to look at the question whether or not a single versus a double transplant is better, and that data has been published recently. And that shows in the US data that tandem transplantation is better than single transplantation. Clearly, there's also been a lot of work in immunotherapy with the introduction of anti-GD2 antibodies. Very important trial done by the North Americans with the introduction of Unituxin. And again, there have been further trials that have also been done in Europe. And also most recently, St Jude's have also shown that you can use anti-GD2 antibody therapy upfront in induction therapy as well. In terms of relapsed refractory neuroblastoma, again, there's been a lot of work around the use of immunotherapy combined with chemotherapy, and also different ways to use immunotherapy in the relapse setting, particularly long-term infusion, which has been done in Europe as well. There's also been an increased focus on actually designing trials for new agents and also to combine those new agents with chemotherapy as well. And so I think it's really important to reflect that really over the last 20 to 30 years, there's been a very successful number of clinical trials that have been done by large numbers of trials organisations that have been influential. And so I think that gives us an important sort of yardstick not only to work from now, but also gives us a great baseline to continue to improve therapy as well. Now, you can see that there have been improvements in neuroblastoma survival. So this is data that Navin Pinto from the Children's Oncology Group published. And again, this is really children with high-risk neuroblastoma who are registered on the Children's Oncology Group biology study. And it's looking at their incremental improvement over a number of years from 1990 up to 2010. And so there have been incremental improvements with improvements in research and clinical trials. But clearly we need to do better. And so one of the things that's also important is that there's been very strong focus in the basic research and also the translational biology. And so that there's been a huge amount of work, which I'm not really going to talk about today. And really what this slide here is trying to show is just really a cartoon that looks at some of the important genes and pathways that we know are important in neuroblastoma, but also important in relapsed and refractory neuroblastoma. And again, many of these pathways have also been found that they can be targeted with specific drugs. And so one of the current translational and clinical challenges in neuroblastoma is how do we translate the knowledge of pathway abnormalities into effective treatments? And again, this is where international collaboration is really, really important. And so on this slide, really what I'm trying to do is just give you an understanding of some of the very powerful sort of research groups and collaborations and consortia that have been put together that are really trying to link basic biology, that are also trying to link that into translational biology and also clinical outcomes. And so if you look at the R2 platform, which is developed in the Netherlands, again, that's a very powerful sort of tool that actually collates and pulls together lots of data in terms of basic research and other research and combines genomic and other data with results. And it provides you with a very easy tool for researchers to be able to look at large data sets of neuroblastoma research and actually look at their the data to look for genes and pathways that are important in terms of their research. So it makes it very easy for collaborators to actually mine data very, very deeply. I talked very briefly about the INRG and the fact that it's led to harmonisation and standardisation of clinical standards for risk assessment and workup. But also the data commons for the INRG is a really important idea because it's actually going beyond what's already been done, but it's then also trying to link the data that's in the INRG to other information that might be available so that you can then try and link that also specifically to tissue that might be banked, genomic data as well. In the US, over a couple of decades now, they've run a program called the PPTC, which is the Pediatric Preclinical Testing Consortium. One of that component is around neuroblastoma. And the idea behind this was really to take some of the resources in research and actually to find better ways to prioritize new and exciting drugs so that they could be taken into clinical trials early on. So part of that platform is at Children's Cancer Institute, but it's got platforms that are also involved with neuroblastoma as well. In Europe, there's the very exciting ITCC P4 pediatric preclinical proof of concept platform. And again, this is a very large scale platform. And the idea behind that is really to then link biologists, translational scientists and clinicians so that you can prioritize targets for which are the best targets to take forward in the disease. Have we got good models to actually look at different targets? Can we also make sure that people are working together? Do we have the right models and the right preclinical drug testing to take that forward? So in Australia, we've been doing a very similar thing in regards to trying to look at personalised medicine and translational research. And so really where I think most groups around the world these days are beginning to work is the idea that we need to do better. But rather than thinking about large groups of patients, we're now trying to focus on the individual patient. And so this is the idea that you have a child with high-risk neuroblastoma or relapsed neuroblastoma, and that you actually want to understand a lot about that child's tumour so that you may be able to identify a better therapy. And so from that perspective, we've been running a personalised medicine program like many other areas in the world. And the idea behind this is that you're using that to help try and find a better therapy, not only for the child, through some genomic profiling, some lab modelling as well, drug testing, but the samples that also get taken from children linked to their clinical data then provide you with a very rich resource so that you also have research reagents to go forward and then actually make them available to do ongoing research as well. And so then it means that we get a better repository of neuroblastoma models and that we can make them available for researchers. And so that if people have got a particular interest in a pathway and we've got a model that's important for that, that model is also available. And so I think this sort of concept of it being trying to focus much more on the individual patient, but at the same time to generate important research tools for researchers going forward is very, very important. And so the personalised medicine program in Australia is called the Zero Childhood Cancer Program. There's a trial run with that, which is called PRISM. But again, I don't really want to talk about that, but again, I want to focus on the idea of interconnectedness and collaboration internationally. So the Zero program is all around Australia, but really, whilst that's important, one of the other really important features of it is the fact that there's a lot of international collaboration that also comes with the personalised medicine program, because we're not trying to do this in isolation, and we realise that we are going to get better results if we actually work together with our colleagues in Europe and North America. And again, then what I wanted to then sort of focus really back around on is this idea that research, it's a collaboration between basic researchers, translational researchers and clinicians, and there's an ongoing cycle of research. And so you don't just stop, you keep on going back and working to try and improve things. And so again, in Europe, between the ITCC and CERPIN, they've got a drug development strategy for many diseases, including neuroblastoma. And so they've just recently published in the last couple of months their second workshop on how to accelerate and prioritise new targets and new therapies for neuroblastoma. So the thing that I really was impressed, and I think this is great about the group, is that it's an international group. Although this is run by the ITCC and CERPIN, they have people who come from all around the world who go to this. And again, it includes clinicians, it involves translational researchers, it involves basic researchers. And the whole idea is to really look at neuroblastoma as a disease and as a problem, identify what are the very high priority targets and how can we as a community work together to actually accelerate improvements in research. And so these are just some of the summaries of the most recent workshop that have been held. So I was going to try and leave it there, but so what I was trying to give you an idea is that there is actually quite a strong interconnected international neuroblastoma network, community. It's been networked for quite a long time now, and it's been happening over decades. It means that, you know, scientists and clinicians learn to speak the same language together, they collaborate together. And it facilitates collaboration that crosses not just basic research, but basic into translational and basic into clinical research. Again, the comparability of trial results means that you get rapid adoption of new advances. And I think this sort of interconnectedness between basic researchers, translational researchers, means that it helps to inform clinical trial design. It also then helps to inform standards of care. And I think also the prioritisation of collaboration between basic translational and clinical research is really important, because it means that we can then focus on the clinical relevance of neuroblastoma biology. It also means that it helps us to identify potential targets for neuroblastoma. And I think it's been a very strong gateway and an enabler for personalised medicine approaches for kids with high-risk neuroblastoma. So that's where I was going to leave it. And at this stage, then I was going to open it up for questions.
Thank you very much, Dr. Traher, for that very informative speech. We do have a couple of questions that have come through. I think the first one would be really useful for just to explain what you mean really with the difference between basic and translational research.
Yeah, so I suppose when basic research is the concept that you might be interested in understanding how a particular gene, pathway or protein works, and that your focus is mainly on that gene, pathway or protein, and you might not be so interested or worried about what other the downstream impacts of that are. Translational research, to my mind, is the concept that there might be an important gene that is the driver of the aggressiveness of neuroblastoma. And as a translational scientist, you might be then trying to work out, well, okay, that seems to be really important in neuroblastoma. How do we target that? Can that be a drug target or a treatment target for a patient? So it's going beyond understanding at the fundamental level and trying to then look at what are the clinical consequences and how that might be used to either find better treatments or is a marker that might be used to identify patients either with a good or a bad response.
Thank you. We have another question which is more specific to older children who are diagnosed with neuroblastoma. Are there key factors present in the genetic makeup of older children diagnosed which are unique to that cohort of patients and which are not seen in younger children?
Yeah, look, that's an excellent question. And again, there's data that's been published on this. And genetically, there are different groups of neuroblastoma. And again, everybody's aware of MICN amplification, and that's generally a genetic change that you tend to see in younger children. And again, in older children, there's been some data which has been published out of, I believe, Memorial Sloan Kettering and St. Jude's. And they had looked at specifically a cohort of older children, and they'd compared them to young children. They were able to find that there were deletions in a gene called ATRX, and that was much more common in children who are older with neuroblastoma. It's very rarely seen in young children. In the sort of middle age group, you do see some kids with it, whereas it's much more common in the older children. And so the ATRX sort of gene is deleted in these patients. And again, it also seems that some of these patients also have problems with maintenance of a mechanism called telomeres, which is the ends of the chromosomes. And again, that's also an important area to think about in terms of targeting neuroblastoma as well.
Thank you. So if there are any other questions that people would like to pose, please do pose them now. I've got a question in terms of, actually one's just come through here. Will we ever be in a position where we can incorporate a new drug to standard of care without having to run randomization or comparative multiple phase trials that take many years, i.e. can we use computer models?
Look, that is an excellent question. And one thing that I haven't really talked about is there is actually a lot of work that's been going on in clinical trials design to try and make trials really efficient and effective. And it's an important question. One of the difficulties is whenever you have a new pathway and a target and a new drug and you think it might work in the laboratory and you need to take it into the clinical scenario, you do need to be able to make sure that that drug is able to be given safely. You also need to be able to get a dose that would allow you to see an effect in children as well. So you do need to do some basic early phase testing in early clinical trials to make sure that it's safe and tolerable. There's been a lot of work by a number of groups around trying to improve trial design so that you can then make sure you get through the process of the early phases of trials very quickly. And again, it's important also to then consider if you have a drug that is effective as a single agent, you also need to then think, can you give it safely with another drug or a combination of drugs as well? And so again, this is one of the important things about safety in trials as well. And as much as we want to go as quickly as possible to get to the point of an effective treatment, it's important to make sure that there's no unexpected toxicity when you put combinations together. In terms of computer modeling for different combinations, in the Research Institute where I work at the Children's Cancer Institute, we do a lot of drug modelling type of work in lab models where we combine drugs. In terms of computer modelling and network modelling, that might be possible. But at the end of the day, when you're looking at treating patients, you do need to have some basic patient information that it's safe and tolerable. I'm not an expert in trials design, but I know the Children's Oncology Group, the ITCC, the University of Birmingham, all their trials units, they've been very careful in thinking about how you design trials effectively to maximise the ability to get useful information in a quick time that's useful for families. But we recognise that does take time.
Thank you. You said earlier, Toby, that there was ATRX as a possible target. Is it actually targetable? Is there a way of actually getting to that?
Yeah, so this is not my area of expertise. I know that their ATRX has been prioritised by the ITCC CEP and neuroblastoma drug development strategy. So targeting telomere maintenance. So the ATRX gene itself, one of its roles is to help maintain the ends of the chromosomes called the telomere maintenance. And so that's been recognised as being an important potential vulnerability in neuroblastoma. And so targeting that pathway generally has been identified as a priority. I'm not personally aware of the specifics of that, but it's clearly an important area. It might mean, though, that rather than targeting ATRX deletion specifically, you're actually targeting other components that are a consequence of ATRX deletion.
Thank you for that answer. We've got one other question that's come up, which is asking if you mentioned, obviously, there are a number of genetic differences within neuroblastoma. Are these going to be taken into consideration? And is the INSS going to sort of update their discussions on this basis and their approaches?
So again, this is one thing I've not put a lot of detail into terms of discussion, but in terms of the risk stratification system that the INRG developed, I mean, one of the things that they actually did do is they started to incorporate much more genomic information. And so again, that's been really important in terms of thinking about non-high risk and also high risk disease, because we do know that there are patterns of genomic changes that occur that have prognostic significance. And so in non-high risk neuroblastoma, sometimes you get gains of whole chromosomes or loss of whole chromosomes. And again, that's a pattern that's usually associated with favourable outcomes. In contradistinction, if you lose parts of chromosomes or gain parts of chromosomes, so small segments, if you gain or lose them, we know that's associated with a poorer outcome. And again, that type of genetic and genomic information, as well as MICAN amplification, has also been included in the way the risk stratification algorithms work, which have been developed internationally through the INRG. Now, it's likely that there will be other changes with time. Nothing should stay static. So where we are now is a point in time, but there will be improvements. And so we know that genetically there are other types of neuroblastoma. So we know that dysregulation of the telomerase maintenance mechanism is important. And again, many of the early publications would suggest that they can be associated with more aggressive disease or poorer outcome. We know that MICAN is also associated with a aggressive disease. And so that as more data becomes available, again, there'll be this iterative process whereby things that have become very important in terms of prognostic classification, they'll become incorporated into risk stratification algorithms. So whilst they may not be there just yet, I would expect that with time, as the data becomes available, it will move into more clinical practice.
Yeah, well, that's great to hear. But one question in terms of collaboration, in terms of the area of personalised medicine programs, are Australia, Europe and the US working together, or are they all doing the same thing?
Yeah, that's a good question. So everybody has a personalised medicine program that's been set up. And so in Australia, the Zero Childhood Cancer Program is the program that runs and drives our personalised medicine program. And that has been running a trial called PRISM. And I think Michelle's probably going to talk a little bit about that. However, as part of that, we have a number of collaborations that actually have been established through that. And so we work very closely with Germany through the DKZF, ZFS. We also work with the Princess Maxima in Netherlands. We've also worked with the Curie Institute in France. We've been working with the Children's Hospital of Philadelphia. We've been working with St Jude's Research Hospital in the United States, as also the University of California in San Francisco as well. And so from that perspective, again, yes, there are collaborations. And so from that perspective, there are also likely to be increasing collaborations with time. One of the things that also happens is that obviously data gets published, the data also gets shared and gets talked about. And so that from that perspective, what we can achieve and do, we'll look at what other results can be attained by other groups, and then look at what we can learn, how we can improve. And so from that perspective, there's a huge focus on personalised medicine programs. And again, there's been a lot of work in terms of trying to collaborate around that. Although what we are doing specifically in Australia has been designed by our team, and our focus has been on genomics, but it's also been on lab modelling. And with the lab modelling, we are trying to then also doing drug screening on patient samples, as well as making animal models, again, for small scale clinical trials in lab animal models to see what treatments may or may not work.
Okay, thank you. We've now got a little flurry of questions. So I'm just going to start with a few relatively quick ones. In terms of the institutions which are working on ATRX, are you aware of which ones by any chance?
Look, I'm sorry, I can't answer in detail. And I know that in our institute, one of our researchers, Talu, has got an interest in the area. We've also got some telomerase research that's also happening over at the other side of Sydney, the Children's Medical Research Institute. But to be honest, it's not an area that I've been following closely in terms of neuroblastoma, so I'd have to pass over.
No, thank you. Yeah, that's fine. We'll move on to the next one, which is, how come targeted therapies are not yet really effective in terms of response as single agents, but still have to be used in combination with already known chemo regimens? Even anti-GD2 antibodies alone are currently mainly used as maintenance when response is already achieved by other means.
Yeah, look, that's an excellent question. And I think sometimes, there are often multiple explanations. I think sometimes we're also just really at the beginning of the journey of understanding how to identify, personalised treatments and targeted therapies for individual patients. And so sometimes also, although we might be able to identify that this is a particular important gene or pathway to target, sometimes the drugs at the moment that we have available aren't necessarily as effective as we would like. And so that there will be ongoing work going on to improve outcome using particular targeted agents. Sometimes it also means that although some of the lab evidence might suggest that this is an important pathway, and if you block that in the lab animal or the lab experiment, that may not work quite so well in the patient, and you might need to look at adding other agents that will allow you to block multiple pathways all at the same time. I think one of the issues that clinicians, which we're concerned about, is that if you're having patients who are being treated on trials or being having access to targeted therapies, you want to also maximise the clinical benefit as well. And so at the moment, again, sometimes combining targeted agents with clinically active chemotherapy allows us to increase the chance of benefit for the patient. And again, it also at the same time, it allows us as clinicians and researchers to actually understand how we can use targeted therapies as well. In terms of thinking about effectiveness of drugs, again, our ALK inhibition is a very important area at the moment, and there have been a number of generations of ALK inhibitors that have been made. And generally it happens that the first time that you make a drug in a class, it may not be as effective as you like, but with further research, you can get more effective inhibition. And so it's likely to happen that there will be some improvements in some drugs. Sometimes also it means that you may not ever be able to have a single drug that will actually do everything that you want. Again, as a disease, neuroblastoma is quite genetically diverse, and there are different sort of genetic subtypes. And it may also be that in a particular subtype, you're just going to have to rely on using more than one type of drug at the same time. Where we are now might be very different from where we are in 5 and 10 years' time. And I think that's the really important and exciting thing in terms of research, is actually being able to take what we've got, but build on that. And again, this is where the international collaborations become very important in terms of understanding how you actually prioritize and actually improve outcomes with specific targeted agents. Again, this sometimes gets back to the basic research and the translational biology, having good models, having good collaboration between researchers to look at the problem and then come up with solutions and ways to actually get better therapies.
Thank you. So a couple more questions before we finish. We have got one question saying, you've also talked about the incredible interconnectedness of the global neuroblastoma research community. Are there still challenges to overcome when it comes to working together in the most effective and efficient way for the benefit of children?
Oh, fantastic question. And look, I think there are always challenges, but I think and one of the real reasons I really enjoy working in neuroblastoma is that I've always had a sense that there is this sense of collegiality and a sense of collaboration. Obviously, research is competitive, so people will compete hard as well, but that's great. And I think, there are obviously challenges to overcome. As researchers, we would always like to believe we could have more resources and more ability to do things. I think in general terms, cancer research, there's a strong focus on it. There's a strong focus in neuroblastoma. I think there are a great number of ideas out there. There are a large number of targets and pathways. And I think, one of the things that and with the final slide that I finished on, which was sort of the ITCC SERP and sort of collaboration to prioritize targets, I think to my mind, that's what's really important is you really do need to have this working together of basic translational and clinical research is to actually work out what we think really is important to make sure that we get the breakthroughs that are likely to have the best impact. I think as an individual researcher at times, you might find yourself you're working in an area that may not be as prioritised as others. And again, I think the other thing is you also need to then make sure that you're looking closely at your collaborative research that you're doing, what results are being published, and again, making sure that, you're working hard. I think funding is always going to be important. I think we're working in challenging times with coronavirus. We're lucky in Australia at the moment that there's a relatively good amount of government source funding through the National Health and Medical Research Council. I think for a lot of charities and philanthropic groups, they are struggling in terms of their donation base at the moment because of the coronavirus. That makes it sometimes harder to get some of the smaller projects up and going. And I think also one of the challenges that we face is we want to be encouraging and exciting young clinicians and young scientists to get into neuroblastoma research as well. But I think one of the really exciting things about the community is I think there are a lot of really exciting young people out there doing research.
Thank you, Toby. Thank you. And you do make it a huge, fantastic contribution to research and obviously care in Australia. We're very grateful to you for that. I don't know if you can answer quite quickly a question we have, which is regarding 1P loss and 17Q gain. Is there anything you can comment on that?
So we were talking before about genetic subtypes of neuroblastoma. We know that classically there are a couple of different types of segmental gains and losses, and so loss of 1P and gain of long arm chromosome 17, they're some of the earlier ones that were identified. And again, they can be associated with a poorer outcome. And so in thinking about non-high-risk neuroblastoma, it gets back to the concept of having either gains of whole gains or loss of whole chromosomes as opposed to segmental gains or losses. And again, sometimes that information about having small segmental gains or losses becomes important in terms of stratification towards treatment. So for some non-high-risk neuroblastoma patients, if they don't have segmental gains or losses and they've got other favourable features, they may have very minimal therapy these days. Whereas if you've got segmental gains or losses, again, those patients are more likely to get more therapy than patients without those features. In terms of the biology behind what's on short arm chromosome 1, long arm chromosome 17, again, that's not really an area that I have any expertise in.
Okay. Thank you very much, Toby. So one last question. Would you say that the childhood tumour cancer research area is as collaborative as the adult solid tumour cancer research area in terms of networking?
Look, that's an excellent question. Look, I'm a pediatric oncologist and I've actually never worked in adult oncology. I do some collaborative research with adult colleagues, and I don't think I really have the expertise to answer that question. I'll just go back to the concept that one of the reasons why I do enjoy working in neuroblastoma is that very first time that I went to the first INRG meeting, I was really struck by, overwhelmed by my international colleagues, and I continue to be that whenever I at a meeting and I meet them, because they are very collegial. They're very good with their time. They're very collaborative. They're happy to share ideas, share data. And again, we've done very well in terms of this idea of being collaborative and working with our colleagues. And so, to my mind, it's overwhelming, and it's great to be working with this community of researchers around the world.
Fantastic. Well, thank you very much, Dr. Toby Trahair, for joining us for the first symposium. We're very grateful for the time you've donated today and answered so many questions. I think we did manage to get through all the questions, so very much appreciate that. We are now actually going to move on to the next session after a very, very short interval, and that session is going to be called Developing New Treatment Approaches to High-Risk Neuroblastoma. So join us back in a minute and we will start the next session. If you head back to the Watch Live page on the event website to start viewing that, that will start shortly as mentioned. Thank you, Dr. Trahair.
Thank you.